methods that we can use to target specific DNA, RNA, and proteins inside of a cell. And so here is a quick overview. Say you want to reduce the levels of a specific protein inside of cells. Maybe just in the lab to see what it does, or maybe actually in a patient to treat a disease that is caused by the making of a messed up protein or making too much of a protein. There are multiple ways that you can go about this because the protein, well, it's kind of just like the end of the assembly line. And so, if the central dogma of molecular biology tells us that those instructions for actually making that protein, the original recipe is written in DNA in the form of a gene that gets transcribed, a copy gets made, this pre-messenger RNA copy. Pre-messenger RNA gets processed through splicing and things to give you a mature messenger RNA, and then those mature messenger RNA copies are used in a process called translation to make a protein. And so if we want to lower the protein, we can go after the protein itself, or we can go after the recipe copies, the messenger RNAs, so that the protein can't get made in the first place. And we can go after the gene so that the messenger RNA can't get made in the first place. But going after the gene is going to be riskier because that's our original recipe. So, except in rare cases, this is typically just being done in the lab. Some of the main methods that we use to target these different molecules, just a quick overview, and then we'll go into the details. If we want to target the DNA, so if we want to target the gene, we're going to use some sort of gene editing tool, something like CRISPR-Cas. And if we want to go after the RNA, we're going to be using something like antisense oligonucleotides or ASOs. We can also use RNAi, so something like siRNA or shRNA. And then if we want to go after the protein, we can use strategies like PROTACs, antibodies, and functional inhibitors. So when we're going after the DNA or we're going after the RNA, we have the benefit of sequence specificity. Because of the one-to-one base pair nature of DNA and RNA, where basically you have one letter that'll pair to one other letter. So you have an A that's going to pair to a T or a U, and then a G that's going to pair to a C. And notice I said A or U, T or U, but that's just because in RNA you have a U and in DNA you have a T, but both pair with A. So you get this specificity in any case, and therefore you're able to directly target specific molecules to specific sequences. And so we can target molecules to DNA sequences, or we could target molecules to RNA sequences based on using these nucleic acid guides. And so in the case of gene editing, we use a guide RNA typically to target destructive machinery or modifying machinery, some sort of Cas protein to a specific DNA sequence. In the case of antisense oligonucleotides, basically these are chemically modified nucleic acids. So kind of like DNA or RNA, but they've got modifications to stabilize them and things. These can bind to the sequence. Some of them just block things like ribosomes. But most of the time and some of them can cause like differences in splicing patterns. So how that messenger RNA, pre-messenger RNA gets processed. And then the main way that you typically see them used, especially in the case of reducing a protein, is by going after and binding the mature messenger RNA and causing it to be cleaved by an enzyme called RNase H. In RNA interference, here you're using an RNA guide, and it's going to bring in a different nuclease, so a different RNA cutter, in this case, Argonaut. And so there's this RNAi pathway, and you can get these guides made inside of cells using shRNA, or you can stick it in kind of like pre-made siRNA duplexes. And then those RNA sequences will be used as guides to take that nucleus, to take that Argonaut to a specific sequence and cut it. And so we're relying on the sequences in all of these different cases to target specific RNAs or DNAs. We can't really do that with a protein. With protein, we don't have that sequence specificity we can use. But something that's some kind of similar strategy where we can target specific things is antibodies, which are little proteins that are going to bind specifically to specific proteins. And this can do things like block binding, or these antibodies can be attached to something else that is going to have another function. So you get like a bifunctional molecule. We can also just inhibit the protein's activity. So especially for like enzymes, so like proteins that are actually doing things, we can do things like use traditional drugs, enzyme inhibitors, things like this to it's like small molecules that'll bind to the protein and just keep it from doing its job. In the case, we can also actually trigger the protein to be degraded using things called PROTACs, so like targeted protein degradation, which PROTACs, or with like molecular glues, where basically we bind a molecule to the protein that's going to bring in a ubiquitin ligase, which is going to add this little protein tag, this ubiquitin tag, onto the protein and cause it to get degraded. So that's a quick overview, and now let's actually go in a little more detail. But if you want more detail, I have much more on each of these in other posts. So let's start by what we mean by how we can go after the gene. So typically this is using some sort of CRISPR-Cas system. And note that when we're actually messing with the gene, we call this gene editing. There's actually multiple types of CRISPR-Cas systems. And so you probably just hear it normally abbreviated like CRISPR. But CRISPR is actually just kind of the part that guides the real action maker. So the real action maker is going to be a Cas protein. So these CRISPR-Cas systems are actually natural bacterial things that they use to target viruses that they've seen before. They kind of like keep a little inventory of sequences from viruses, and then they can use those as guide RNAs to direct Cas proteins to those sequences. And so we can take advantage of this in the lab by using kind of modified versions in order to introduce guides that will then direct Cas proteins to specific DNA sequences. Now, there are different Cas proteins that can do different things. The kind of conventional or original Cas protein that we think of that's really good for like knocking out specific sequences is going to be Cas9. It's going to take an RNA guide and recognize double-stranded DNA with the matching sequence and then cut it. And what's going to happen is then the cell's going to try to take over, but it typically does a pretty bad job. So it has, if you don't give it something to try to put it in instead. Basically, you can try to stick in another sequence if you want it to get like swapped out for something or changed. But if you don't want to give it another sequence, then it's going to kind of just try to piece things together as best it can. And this often leads to a non-functional protein. And so this is a strategy we can use for genetic knockout, where we're totally reducing the making of the protein, by reducing the making of the messenger RNA and therefore the protein. There are different Cas proteins, though, and other ones will do different things. And some of the Cas proteins are also being engineered so that they can be more specific. You have ones where, say, you might need, so Cas9 cuts like both strands of the DNA. There are also Cas proteins that'll only cut like one strand, and so you need two guides to actually make it so that, to make the sequence double-stranded, to make a full break. And this can add a little more specificity so you don't get off-target activity. There are also like prime editing is a growing thing where basically these are going to send in machinery that can actually make changes to the sequence with like reverse transcription and things like actually at the site of where you want things changed, some really cool stuff like that. And then there's also like base editors that can do things like cytosine deamination and things like this to change just specific letters, so not without making any actual cuts, but just kind of like swapping out which letter is there by kind of messing up one of the DNA letters so that when it gets copied, then the sequence gets switched. And so that could be used to make specific like single nucleotide changes. But in the lab, we often use this just like Cas9 strategy in order to to like defunctionalize a specific gene. But you can also try to get homology-directed repair, where you stick in a sequence to get it swapped in for that alternative. So that was targeting the gene itself. Now let's talk about targeting the RNA. And the two main methods I'm going to talk about are these antisense oligonucleotides, or ASOs, and RNA interference, or RNAi. And I have much more on both of these in other posts. But as I mentioned, both of these are going to rely on using nucleic acids. So one of the things about the CRISPR system is if you want to actually use it, you don't just have to introduce the guide, you also have to introduce the Cas protein. And this can actually be a much bigger hurdle, and especially if you were trying to do it inside of like patient cells, or like, so often when we have like these treatments for various diseases, sometimes they're done like in cells that are taken out of the body, like bone marrow, and then like put back in, things like this. But if you want to actually treat cells like throughout the body, well, now you have the challenge of trying to get in a whole protein as well as the RNA. There are things like trying to make smaller versions of the Cas proteins. So there's a lot of work being done, and I'm not going to go into. But basically, with the CRISPR-Cas, you have to get in the RNA, the guide, as well as the protein. When you're talking about these strategies targeting the RNA, here we just have to get in the guide. And so with the antisense oligonucleotides, what we're sticking in is going to be a chemically modified sequence of nucleic acids that's going to be antisense to the messenger RNA. So basically, the sense strand is going to be the strand with the information in the for making the protein, and then the antisense strand is kind of like the matching sequence, the other strand that you don't really focus on in that DNA. And so that other strand is antisense, so it'll bind to that sense strand, it'll bind to the messenger RNA. And we designed it in such a way that it's a short sequence, so short, so few oligonucleotides, so few nucleotides, a few of these nucleotides. And these nucleotides are kind of like modified versions typically of they're like DNA or an RNA, but they've got modifications in order to make them more stable. We also often put modifications on for like the RNA guides that we're using for the RNAi as well. Modifications commonly include like changes to the 2' OH position to prevent the RNA from like attacking itself or from being vulnerable to nucleases, as well as backbone modifications that also help with the nucleases. And these also help kind of like the molecules survive in your body without being seen as foreign. We can also have modifications that are going to target nucleic acids to specific tissues or cell types. A common example is like GalNAc conjugation, where you put the sugar chain on that then gets these molecules targeted to liver cells, which have the receptor for that. And we can get all these in using various methods. So we call like transfection when we get nucleic acids into cells. This can be done with various ways. For talking about into like live cells in the body, we can use things like lipids. Lipid nanoparticles are a big thing. Cationic polymers, so positively charged, long positively charged molecules that can kind of neutralize that negative charge of the nucleic acids. There are viral delivery methods, things like adenoviral vectors or adeno-associated vectors, AAVs. Antisense oligonucleotides, so these ASOs, can be designed so that they bind the pre-mRNA or they bind the messenger RNA. If they bind the pre-mRNA, this is commonly used to do things like alter splicing patterns. So splicing, remember, that's the process where you're kind of processing that pre-mRNA into mature RNA. And this happens by removing these regulatory regions called introns. In various diseases, you can get problems with that removal that causes them to be spliced together incorrectly. And these ASOs can be designed so that they kind of hide the mutations, or they kind of bring in the splicing machinery, keep away the splicing machinery so that those transcripts get processed correctly, they get spliced correctly. And this is the basis for drugs like Spinraza for spinal muscular atrophy. So there's already examples of this being used in the clinic. Other ASOs target the messenger RNA. And so this can do a couple different things. One is it can kind of just sit there and block things, like block the ribosome, which needs to use the instructions to make a protein, and now there's an ASO bound where it's supposed to be. But the main method it works on messenger RNA is by causing there to be this like RNA DNA hybrid that's a substrate. So it's something that can be cut by this RNase H. And so RNase H will see this hybrid between this ASO and the RNA, and it will cut the RNA and cause its degradation. You can also design ASOs so that they're like bifunctional, kind of like what we talked about before with the PROTACs or with the antibodies. Basically, you can have these ASOs use that sequence to target a specific RNA, and then also bring in something that's attached to that ASO. The other main strategy using to target the messenger RNA is RNA interference, or RNAi. Here you have a short RNA guide that's going to bind to a protein called Argonauts. This is a protein that's naturally in our cells, so we're not relying on having to bring in a whole nother machinery like with the Cas system. Instead, our cells use RNA interference to regulate our own genes, typically through a form called microRNA mediated regulation. And I have much more on that in other posts. But if you stick this guide, you can design this guide so that it's going to target a specific sequence. AGO will bind to this guide. AGO will go to that sequence, and AGO will cut the sequence. And then this will expose naked ends of the RNA that can then get chewed away. So normally in messenger RNA, it's protected at its ends by a cap and a tail. But when you cut it in the middle, well, now you're exposing raw ends that can get chewed away. And so this happens with the RNA interference from cutting by Argonauts, and it happens in the case of your ASO by cutting by RNase H. And both of those are nucleases that are already in your cell, as opposed to the Cas proteins. But how do you get the guide into AGO? There's a couple different strategies. So AGO is going to use a single strand, but it loads like a duplex. And so you can give it this duplex directly, this like small interfering RNA duplex siRNA, or you can kind of give it a precursor, these short hairpin RNAs, which then get cut by dicer to form basically a duplex that can get loaded into AGO. In terms of this shRNA, what's the benefit of it is that you can actually get these cells to make it in themselves, so you don't have to keep giving these cells more and more of the siRNA. So you can either stick a plasmid in there that has the instructions for making it, and the cells will make it. But then as those cells divide, that plasmid will get diluted out, so this gives you a transient transfection. Or what you can do is you can kind of get it stably integrated into the genome. And so then it's going to be permanently passed down and expressed, which will give you long time expression of that to give you a longer knockdown. So we typically refer to this as a knockdown when we're reducing the levels of the messenger RNAs, as opposed to knockout, which is like what we do with CRISPR, where we're actually going after the gene and permanently permanently eliminating the production. And so these strategies in terms of like actually doing the plasmid or doing the transfection, stable transfection, this would typically be in cell culture, but you can provide siRNA complexes, or you could provide the shRNA complexes into cells like in the body. And so there's actually RNAi therapeutics in use. So we've gone after the DNA, we've gone after the RNA, and now we're going to go after the final product, the protein. So one of the downsides about going after the protein is that, except in certain cases, you lose the specificity for the mutated version. So in the case of when you're going after a sequence, like you're going after a gene, or you're going after the mRNA, you can actually go after the part sometimes that has the mutation. So you can target just the mutated version. You could reduce the production of, say, the mutated messenger RNA, but not the normal one. And this would then reduce the toxicity because you're still going to have the good stuff made, but without the bad stuff made. Assuming, of course, that this is in a heterozygous individual. But unfortunately, often what happens is that you have or I guess unfortunate for this case, but fortunate in the sense that you would need two bad copies, is that a lot of the genetic diseases are homozygous recessive, you have to be homozygous recessive, which basically means that you need two bad copies of the gene in order to have the disorder. So therefore you couldn't selectively target a good one. But there are some diseases that are caused by mutation in just one copy of a gene, and therefore like some of your messenger RNAs are still going to be okay, and so you can go after the bad ones but not the good ones. And so there's various strategies that are used for things like that. But in the case of a protein, it's going to be harder to target just a mutant one, although there are cases such as with like KRAS inhibitors where a mutation actually makes it so that you can make specific the molecule looks different enough, it has different features that allow the drugs to selectively target the mutated version. But anyway, how do we actually target proteins? There are various strategies. One is a growing area of research is like targeted protein degradation. And so this can be done in a couple different ways. The main strategies are going to be PROTACs and molecular glues. And both of these, what they're going to do is they're going to bring in ubiquitination machinery. So like ubiquitin ligases. Ubiquitin is this little protein and that can get stuck onto other proteins in order to target those proteins for degradation. So these proteins, ubiquitin ligases add ubiquitin to proteins, and then the ubiquitin binds to a proteasome, which is like a protein shredder, and the protein gets degraded. The idea with a PROTAC or a molecular glue is to bring in the ubiquitin ligase to specific molecules so that you can tell them to ubiquitinate the protein and then get the protein degraded. And so with a PROTAC, basically, these are proteolysis targeting chimeras. These are heterobifunctional molecules, so they have two different parts and do two things. One part's going to bind to that ubiquitin ligase, and another part is going to bind to the protein of interest, and then they're connected by a linker. And so PROTACs, basically that part that targets the ligase, there's some common ones that can be used, and you can kind of like swap out the different parts that bind to the protein of interest, but use the same part that binds to the ubiquitin ligase. And so people try to determine parts that bind to the protein of interest, and then kind of connect that to a set of known parts that will bind to ubiquitin ligase, and then try to figure out how to optimize the linkers and how to optimize the binding and all sorts of stuff. It's not as easy as it looks, but you can bring in the ubiquitination machinery. Molecular glues are kind of like harder to design. An example is like the thalidomide, and basically what it does is it kind of is a single molecule, so it's not that like bifunctional, it's not that heterobifunctional or whatever. I guess not technically, I don't know, it doesn't have like two parts. Instead, it's got a single part that's going to bind to the protein of interest and bind to the ligase and kind of bring them together. And yeah, sometimes they're called IMIDs, but yeah. PROTACs are the ones you hear more about. They're easier to kind of like predict how they might form. Other methods include like antibodies. So a lot of those biologics drugs you might hear of. Basically, a biologic is going to be anything that's made in a cell or in an animal or something like this, as opposed to your conventional small molecules, which are things just like your Tylenol, your Advil, all that sort of thing that are made through synthetic chemical reactions. Commonly, so biologics, they're often they're often proteins, and they're often antibodies. And so basically, a common example you might have heard of is Humira. Humira is a monoclonal antibody. So what's an antibody? It's a little protein that'll recognize a specific thing. And a monoclonal antibody means that all of the antibodies in that vial you get are exactly the same. And so they're all the same antibody, as opposed to a polyclonal, where you have a mix of different antibodies that might all recognize the same thing, but the antibodies themselves are different, and they're going to recognize slightly different locations on that thing that they're recognizing. And in the case of Humira, what it's going to be doing is it's going to recognize an immune signal called TNF alpha, and it's going to bind to TNF alpha and prevent it from causing overactive immune responses. So antibodies can bind to specific molecules, specific proteins, and block the binding of other proteins, hide specific sites, things like this. There are various ways that antibodies can be designed to target specific different proteins, and these antibodies can also be linked or conjugated to other molecules to bring other molecules, maybe other proteins, into specific proteins. And so antibodies are kind of like the equivalent of a sequence for in terms of targeting a specific molecule, except that it's more complicated than that. But I mean, like in the lab, if we want to detect a protein on a Western blot, we use an antibody. And if we want to detect a DNA or an RNA on a northern or a southern blot, we use a radio-labeled or fluorescently labeled sequence probe. And so that's basically, yeah. In the lab, you kind of think of antibodies as sort of the equivalent of the sequence, but you can't just design an antibody in the same way that you would design a sequence that matches. Instead, it's more just like trial and error and trying to get ones that actually match. And they're not going to be quite as specific. Finally, we have just like your functional inhibitors that block the activity of the protein. And so when we talk about protein binding partners, we call these things ligands. And in the case of an enzyme, so in the case of a protein that's actually causing some reaction to happen and speeding up or catalyzing a reaction, basically with an enzyme, we call the thing that it's actually changing their substrates. And so the substrate will bind to the active site of the protein and then be changed. And we can design molecules inhibitors that actually bind to that active site, and they can actually like pretend to be the molecule, and they can actually like compete for the binding site, or they can bind to the protein and kind of just like defunctionalize it. There are covalent and covalent inhibitors that will actually bind more permanently to these sites. This is one of those cases where with the KRAS, if you have the mutation actually introduces there's a mutation that mutations actually introduce sites that make it so that you can get covalent inhibition of the mutated version, but not the normal version. And so check out Keyvan Shokat's work on this. It's really, really fascinating and life-changing. But yeah, so we also have some of the diseases are caused by overactive or excessive receptors. So these are proteins typically in the cell membrane that are going to bind to some sort of signal and cellular signal, and then therefore they're going to kind of like change shape or something that's going to relay that signal to through other molecules. We have antagonists, which are basically molecules that will bind to the receptor and prevent their activation. And so some drugs that you see, some ways to target the activity of a protein, are going to be classified as antagonists. So to review, we can target molecules at multiple levels. If we want to target DNA or RNA, we're going to use the sequence-based method, taking advantage of the sequence-to-sequence complementarity. So if we want to go after the gene, we'll use a gene editing tool like CRISPR-Cas. which uses a nucleic acid guide to guide a Cas protein to a specific sequence. And depending on what that Cas protein is, it can then cause that, cut that sequence, cause it to be knocked out or permanently disabled. You can have it swap some sequence in. There are also proteins that will do things like change single nucleotides, as well as the prime editors that will actually change the sequence there in place. And those are kind of new and upcoming technologies in the works. If we want to go after the RNA, we can use antisense oligonucleotides, or ASOs, which are short chemically modified nucleic acids. So they're chemically modified to be more stable, and things like this. They can bind to the pre-messenger RNA and alter its splicing patterns, or they can bind to the messenger RNA, block things like ribosomes to prevent translation, or more commonly actually cause there to be these ASO RNA hybrids that get cut by RNase H. If the RNA interference, you're using RNA guides, such as from shRNA or siRNA, in order to direct a different nuclease argonaut to a sequence in the messenger RNA and cause it to be cut. So with either of those, you can cause the degradation of the messenger RNA and therefore prevent the production of protein. And finally, you can go after the protein itself, such as with PROTACs, which are going to send in ubiquitin ligases to a specific protein, cause that protein to get tagged for degradation. You can use antibodies, which are going to block the binding of the protein to its normal partners. Or you can use functional inhibitors that are going to go after, such as like going after the active site of an enzyme to block its activity. So hope that helps you understand some of these different techniques that we can use. And I have more extensive posts on all of these things.